Very much. It is a pleasure to be here. Thank you for the introduction, Marcel. Before I will start my today's presentation, I would like to tell you that I'm honestly, genuinely proud of the fact that I am part of the NF Core community. In my company, we have motto Code Against Cancer, which really sits in my values, and it motivates me and reminds me that our work can actually change the world. And as we are usually a little bit further down in the process of drug discovery, it is easy to forget that our hard work is directly connected. And please, be proud of your work and remember that we are an essential part of the drug discovery process. Today, I will be speaking about one of the projects which we realized on the behalf of Boehringer Ingelheim, which is focused around integration of Limma VOOM package into NF Core differential abundance pipeline. I will start from the quick introduction. I will tell you a bit more about our company, about the purpose of the project, how we manage that, and what will be the future of this integration. And finally, I will make a quick summary for you. So, the introduction. Ardigen is an innovative AI CRO, which is a part of AI in drug discovery market. And our promise is to increase the probability of success of the drug discovery process and the acceleration of that process. We are nowadays 9 years on the market, and we are proud of the fact that since the early days of our company, we've been working very closely with machine learning approaches, and we've been integrating them into our projects. We succeeded over 400 projects nowadays with over 100 partners. And what is quite important here is that we regularly publish and co-author scientific publications. Ardigen enables digital transformation and unlocks full potential of the data. And we position data in the center of the operation to unlock its full potential, because nowadays there's a ton of data which can be used and leveraged in the science. And we start from the robust infrastructure and accessible data universe. And finally, we end with the layer of actionable scientific insights. So what is very important in our company is that we are able to successfully work with these 3 layers and to efficiently generate scientific insights in the process, from the target identification up to clinical stage. And, you know, bioinformatic workflows are just a small part of our offer. However, in my opinion, as a person who is, very into Nextflow, they are essential in drug discovery process. I like to compare them to the organism, and which operates and connects everything with blood vessels, which are transferring blood. And in the drug discovery process, bioinformatic workflows are doing exactly the same job. They're transferring data through the drug discovery process. And a few words about bioinformatic workflows in details how we work. We support our customers in 4 stages, I called it, with pipeline development, where we design the pipeline from scratch or move it into production. We assist with deployment and all kind of migration, almost exclusively to Nextflow. And we support automation and data management efforts of our customers. And finally, we frequently help with results interpretation. So now let's talk about the project, and I will start with small background. Differential abundance pipeline can be used to analyze data represented as matrices, comparing groups of observations to generate differential statistics and downstream analysis. The pipeline supports RNA-seq data, which are most frequently generated by RNA-seq pipeline. So now let's take a step back and talk about RNA-seq data in clinical space. I will skip the definition because, well, nowadays it is quite obvious, and I can bet that you know what the RNA-seq is. However, I would like to mention the example applications. And those are disease mechanism studies, biomarker discovery, personalized medicine, therapeutic development. And, it might not be very scientific measure. However, the RNA-seq pipeline is one of the, well, not one of the, it's the most popular NF-core pipeline. So that can be important in next topic, which is related to challenges of the data which origins from clinical studies. And first main challenge is the complexity of clinical data set, where the studies often involve diverse populations, varying data sets, and very complex treatment schemes. And second is incomplete paired patient data, where missing data can bias results and significantly reduce statistical power of the analysis. And third one is related to data integration and interpretation, where accurate analysis requires robust statistical methods to handle variability and ensure reproducibility. So what is Limma VOOM, and how we found it very useful together with our customer. So Limma is a statistical package which was originally developed to analysis of microarrays. And VOOM is extension which was developed to add a support for RNA-seq data in Limma. And VOOM transforms the count data to log counts with associated weights, what allows the use of linear modeling, which is the main point of Limma. So the key features of this combination together is robustness, flexibility, and speed. And it can effectively deal with complex experimental data sets, which are including paired samples or multiple covariates. And the main strength is ability to handle very well the incomplete paired data by using linear modeling, and the support for significantly more complex model designs which are not necessarily supported in other tools. So now I would like to have a quick comparison with DSeq. DSeq is also a very widely used tool for RNA analysis of RNA-seq data. And main key differences between those two are statistical methods, how they handle missing data, and the normalization methods which are implemented. So DSeq focuses more on the, when it goes to statistical models, on count data, where it is using negative binomial distribution. And Limma VOOM is using the linear models, which are a little bit more flexible when it goes to handling missing data. And when it goes to normalization, Limma uses, mentioned earlier, VOOM transformation, versus DSeq uses the size factor normalization and variables stabilizing transformation. Sorry. So how we achieve this integration? First, we had a very long discussion with the moderators of NF-core differential abundance pipeline, and it ended up with very, very huge simplification of our project. So huge thanks to you guys. In the end, we ended up with a updated NF-core module, which is responsible for Limma differential analysis, where we created new environment with additional packages, and we introduced additional parameters which are allowing to handle well new logic, and also new pipeline. In the pipeline, we extended visible here, green path, which is responsible for RNA-seq data flow, which is nowadays very well handled also by Limma in the pipeline. So if you would like to use this, right now this is on dev branch, and we added 2 new profiles in the pipeline. One is called RNA-seq Limma, which contains default parameters for this analysis, and also there is a test RNA-seq Limma profile, which is executed with the example test data, which are stored in the repository. And what next is the extension of the Limma VOOM module to handle even more advanced mixed models. And this is currently in this stage of the tests implementation. And next will be the incorporation of IHW correction. And that, I hope, will be executed in probably upcoming few weeks. So very important here is our team and the NF Core community and our customer. I would like to mention Thomas Schwarz, Alexander Pelzer, and Ramona Schmidt for granting us the opportunity to work on this project. And I would like to also thank you to Oscar Wacker and Jonathan Moning for the hard effort which you put it into development of this pipeline and very long discussions with me during the implementations. And also on our end, I would like to thank you, Ola and Michał, for the bioinformatics support which you gave me during this project. Thank you for the opportunity to be here, and well, if you would like to hear more about this project or Ardigen, step back next to our booth.